Hello students, and welcome to our recording of the cell respiration lab. Alright, so what are we going to do in this lab today? We're going to measure the rates of cell respiration in germinating and non-germinating beans. Alright, so what does that mean? Well, we have, get this out of the way. We have two kinds of beans. Well, we have pinto beans, and we've been taking these pinto beans and we've soaked them in water, and they're starting to germinate. Now, germination is just a fancy word for that the seeds are starting to grow. Here is your lab. This lab can be found in the class handouts section of chapters 8 and 9 in the Schoology folder. This is the lab and this is the introduction. So what you want to do is you want to briefly read through this introduction and then pause the video from time to time because here is some information that you want to write down. Now, ultimately, what we're studying is how fast does cell respiration take place inside a bean. Now, ultimately, cell respiration is an energy converting chemical reaction. Really, if we had to summarize chapters 8 and 9 in 2 words, it would be energy transfer. Chapter 8 is all about the transfer of light energy into chemical energy through the process of photosynthesis. Chapter 9 is about the transfer of chemical energy form 1, glucose, into chemical energy form 2, ATP. We're going to measure the rate that this chemical reaction happens. How fast does this chemical reaction happen by quantifying or measuring the amount of CO2. If the amount of CO2 is going up, then we infer that this reaction is happening. If the amount of CO2 is going up really fast, we infer that the reaction is happening fast. If it's going up really slow, we infer that the reaction is happening slow. So that's the basic premise of the lab. All right, so in this space right here, you want to write down the chemical equation in both symbols and words. So the molecular formula as well as the labels underneath it. We need to know that equation for the exam. And then in this space right here, we want to write down these few objectives. And these objectives are listed right here. So you can open up the lab. Once you get the lab open, you can go ahead and pause. And then again, if you need to back up, you may. But record this equation here, and then record the objectives here. All right, once you have this open, you can get that done. We don't need this open anymore for now. So I can minimize this, right? And we'll go to the big screen. There's our objectives. I'm going to move myself over here because now we want to take a look at the lab. How are we going to measure how fast this CO2 is being made? And that takes us to this graph right here. So what I'm going to use, I'm going to hold this up, it's very little, but this is called a CO2 sensor. These little tiny holes pick up CO2. They measure the amount of CO2. And then I've got my male gene respiration, oh, not that one. Got my male gene respiration chamber. And I'm going to put 25 beans in there. So 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 21, almost there. There we go. So now I have 25 beans, 25 germinating beans in here. Now I'm going to stick the CO2 sensor in here, and this sensor is going to start collecting the CO2. Now there was some residual CO2 inside the container, so we got to let it sit for just a little bit of time because we want to. What we're measuring is the amount of CO2 in this going to go up? Is it going to go down? Is it going to stay the same? And the rate at which that CO2 is produced, if it's produced at all, is an indicator of how fast that chemical reaction, cellular respiration, is taking place. All right, now as you can see down here, these are numbers. CO2 is measured in parts per million, and I'm going to hit the word collect. And then now we'll start to see that our data here is collecting. I'm going to change just one thing here as it collects. Stop for a second. So like this. We want to change this to 300 seconds because we don't need it to be going for 10 minutes. And let's collect. It's going to clear that out and we'll go from there. So now we have this set for 5 minutes. Notice I just want to point out as the lab runs, as the test runs, here's our CO2 gas, right? So this is a change over time graph. Here's the time, right? So what change do we see? We see a change in the amount of CO2 that is being produced. Note here that CO2 gas is measured in ppm. That stands for parts per million. It's an actually remarkably tiny percentage of the atmosphere that is actually carbon dioxide. Think about in a breath of air, 80%, almost 80% of that breath of air is completely inert nitrogen. Around 18-19% of it is oxygen. There's so little CO2 in the atmosphere that it's measured in parts per million. Right now, typically there's about 406 to 407 parts per million CO2 in the atmosphere. That means that if you took a breath of air and divided it into a million pieces, 406 of them would be carbon dioxide. So not very much at all. All right, so as this runs, you can see, so this is set to run for 5 minutes. And as this runs, the CO2 is collected, and then we can add some information to it as it runs. All right, so for instance, I'm going to just click on this, and I'm just going to put add annotation, and we're going to title this room temperature germinating beans. We're really doing 2 experiments here. We're going to run 3 tests, but we're doing 2 experiments. We are going to compare the rate of cellular respiration in germinating beans to the rate of cellular respiration in non-germinating beans. And then we're going to compare, using these same beans, we're going to put these on ice. So we'll compare the rate of cellular respiration in non-germinating beans. We'll compare the rates of cellular respiration in germinating beans at room temperature to the rates of germinating beans at cooler temperatures. What happens if we slow or cool those temperatures down, right? Again, there's our little sensor as it's collecting its data. All right, so we don't need to sit through this whole downtime as we wait for the data to collect. So I'm just going to minimize this, and then pause this for just a second. Nope. All right, students, so all I really did was I just let the test complete its run. So it ran for 5 minutes. We just paused it for a couple of minutes to let it finish out. Now, as you can see, if we take a look at the graph, here we've covered a span of 300 seconds or 5 minutes. And then I put in this label, the room temperature of germinating beans. So this is the graph. You can see the obvious pattern. And note that down here, I've got this 6.90 ppm per second. So what this is telling us is that every second, the amount of CO2 that was available went up in 6.9 parts per million per second. So every second there was almost 7 parts per million more particles of CO2. So clearly we see that germinating beans do go through cell respiration. All right, so now what I'm going to do is I'm going to take this sensor off and I'm going to let it recalibrate. I'm going to take these beans, and I'm going to get them cold. So I'm going to get these beans and I'm going to put a little ice in here. So I'm just dropping a couple ice cubes in here. And then I'm going to put in just a little bit of water. And then I'm going to take this and I'm going to sit this on ice. So now we're going to take a few minutes and we're going to let those beans get really, really cold. We're going to let those beans get really, really cold, and then we're going to test the germinating capability of those beans at cooler temperatures. What I've also done is in here I have 25 non-germinating beans. So now we're going to test the rate of cellular respiration on these non-germinating beans. Now I got to recalibrate the sensor just a little bit, so I'm just going to fan this down until the number gets, well, basically it's got to get below 800. And as I do this, you can see that CO2 is dropping in the little lower right corner of the graph there. So now as we do this, I'll get started on collecting our second data set. Alright, so now we're going to let this sit for another minute to let any of the residual CO2 that's in there get used up, grabbed up by that sensor. And then after a half a minute or so, we will start collecting again, and then we're going to see a second data collection set. And then as this runs, we'll let that run for 5 minutes, and then we're going to do one more test. All right, so as we do this, and you fill out the lab and you fill out the questions, keep in mind the big picture. Ultimately, what we're doing here is we are looking at how fast cellular respiration is happening in these germinating beans. We really have 5 components, right? 2 reactants. We have glucose and oxygen going in, and we have water, carbon dioxide, and ATP coming out. So how are we measuring how fast this reaction is happening? Well, we're using a CO2 sensor. So we're measuring the amount of CO2. So in that chemical reaction, if a product is being increased, that means the chemical reaction is happening and the reactants are being converted into products. The more product that is made, the faster that reaction is happening. All right, so let's go ahead and collect our data. So now we're going to see that a second set of data is going to get started. It's going to start running. It's still going to run for another 5 minutes. You can see there's the line right there. What I can do is I can pull this over a little bit, and then just by way of comparison, we can show that first data set. All right, so here is our first data set. That was our room temperature germinating beans. Now these are our room temperature non-germinating beans. And within the first half minute or so, you can see there doesn't seem to be a whole lot going on. Hopefully that doesn't surprise us. Hopefully we would think that, well, maybe those beans are using a little bit of oxygen, but we know that essentially non-germinating seeds are not growing, so they wouldn't be using a whole lot of the glucose that's in that seed. Remember, in a seed, a portion, only a tiny portion of the seed is actually the plant. Only a tiny portion of that seed is actually a plant. Most of what's inside this plant is called endosperm. It's essentially the glucose that is the food that's stored for the baby plant as it grows. As this baby plant grows, it can't photosynthesize yet because the leaves are not big enough. But once that plant emerges from the ground and the leaves can start photosynthesizing, well, at that point, all the glucose, all the starch, all the nutrients, all that yummy goodness that's inside that seed, that has been burned up. All right, once again, we don't need to sit here and just let this run for the whole time, so I'm going to pause it, let it run, and I'll be back in a couple minutes. All right, students, well, now we had the second test run, and as you can see, this blue line is the indicator of how much CO2 was being produced in our non-germinating beans, right, in our non-germinating beans. All right, so as you may have suspected, not a whole lot going on there. So you can see this line is basically flat. So what this tells us that over 5 minutes, we had basically no change in the amount of CO2 that was inside this container. We would infer from that that the chemical reaction of cellular respiration is not taking place. the glucose inside the seed is not being converted into water, ATP, and carbon dioxide. So again, always keep in mind that big picture. All right, so now we're down to our final test. We got to do one more exam, or one more test, one more trial. Now, what we need to do is we need to test the beans that were sitting on ice for about 8 or 9 minutes or so. So I want to blot these beans dry because I don't want there to be any excess water in there because that can screw up my ratings a little bit, my readings. So I've taken the original 25 beans that we had, that we ran and gave us this rate of CO2 production. Now I'm going to take those same beans that have been sitting on ice, and we're going to reset the sensor down to a good number. We're going to put this in here. We're going to let this start collecting data again. Only now we're going to make sure that this stays on ice. We're going to make sure this stays on ice. So now we're going to germinate these beans. We're going to record the CO2 production of these beans for 5 minutes as they sit on ice. All right, so remember, this is really our trial 3, right? We have beans that are germinating at room temperature, beans that are not germinating at room temperature. Now we have beans that are germinating at cold temperatures. So let's go ahead and hit collect. And we'll just observe the data pattern for, maybe a minute or two, and then we'll go from there. Now every time I hit collect, it clears out the previous runs, but what we can do is we can go over here, we can take a look at this one, we can take a look at that one. Right, so now we can see, now we can change this data, rename this one, we'll call this germinating at cool temps. Germinating at cool temps, and rename. Now it does state in your lab to record the temperature. I recorded those here in my basement, so it was 20 degrees Celsius. That's the room temperature. So the room temperature of the germinating beans was 20 degrees, and then the cool temperature was 6 degrees Celsius. All right, so now you can see, right, here's our pattern. We'll see where this takes us. We're somewhere between about 75 seconds, so almost a minute to a minute and a half. We'll let this go for another 3 or 4 minutes, and then we'll be back. All right, students, I'm back. We can go back to the graph. Now let's take a look at our final product. So we ran our 3 tests. The 3rd test is finished. I've gone ahead and done the annotating and figured out the slope, the rise over run, while we were paused, so you don't have to sit through me just figuring that stuff out. All right, so here the software does the graphing for us. Here is our room temperature germinating beans, our rate of 6.9 parts per million per second. So every second it was making 6.9 parts per million more every second. All right, so this middle line is our cold temperature germinating, so you can see it's obviously much slower. And then there's our slope. Remember, slope is rise over run. Since this is change over time, it's the rise in CO2 over a certain length of time. So every second, how much did the rate of CO2 production go up? Clearly this one's the fastest. The room temperature germinating beans significantly slower, and then the the cold temperature germinating beans significantly slower, and the room temperature non-germinating beans, right, literally 0.0 parts per million per second. So clearly the non-germinating seeds are not going through cell respiration. All right, so what are some of the factors that affect the rates of cell respiration? These are some of the questions that you're going to address. All right, so if I go to this now, I'm going to make this a little bit bigger. This is the handout that you need to fill out. Again, this can be found in the documents or in the class handouts folder of chapter 9. We've recorded this temperature at 20 degrees Celsius. We've recorded this temperature at 6 degrees Celsius. So that's that data for that. I just want to get it centered. No, where are we here? I guess we don't matter. There we go. All right, so that's our data temperature-wise. What's our rate of cellular respiration? What's our parts per million per second? Right, you can get that off of the graph. The graph is also saved. Right now that the lab is done, you've seen how it's done. You can access the graph in the class handout section as well. And then be sure to give yourself some time to answer these analysis questions. Pay careful attention to questions 8 and 9 when you're asked to identify the independent variable and the dependent variable. Remember, we did 2 experiments here. Really, we ran 3 trials, but that gave us 2 experiments. We compared germinating at room temperature to non-germinating at room temperature, and then we compared germinating at room temperature to germinating at cold temperatures. So be sure to be careful and thoughtful when identifying those independent and dependent variables. You don't have to worry about number 10. If we were doing this in the classroom, you'd actually create the graph, but you don't need to do that. You just need to do the data analysis from the graph for the lab. No need to add a picture of the graph to the lab. All right, I hope that was helpful. Again, as always, keep in mind the big picture. We're measuring the rate of cellular respiration by quantifying the production of one of the products, CO2 in this case. I hope that helped.